Lab 7 Enzymes by Emma and Anthony, and this presentation consists of our introduction. Let's begin by learning about enzymes. So, what exactly are enzymes? Well, enzymes act as catalysts to speed up chemical reactions within cells, and enzymes are also protein, and they are usually pretty large molecules. Enzymes contain an active site, which is a location on an enzyme where the chemical reaction takes place. And they also contain a substrate, which is the reactant molecule that binds to an enzyme at the active site and participates in the chemical reaction. And this is often referred to as an enzyme substrate complex. So let's take a look at the diagram over here. We can see that this is the enzyme and its active site here, where it has empty grooves, it is where the substrate will bind. And enzymes bind their substrates with a very high degree of specificity. So it's almost as if the enzyme would be a lock and the substrate would be a key that fits into it. So not any substrate can just come along and bind to any enzyme. They are very specific to certain enzymes. So certain substrates can bind to certain enzymes. Now to move on to the induced fit theory. This theory states that an enzyme will catalyze its reactants to a product only after the conformational changes between itself and its substrates take place. So what this means is that ultimately substrates will bind more tightly to an enzyme and the chemical bonds will be strained. So this means that ultimately the substrate and the enzyme will be binded much closer together and more tightly. So let's take a look at the catalytic cycle of an enzyme. So here we can see this very easy to see diagram. So the first thing that happens is we have an enzyme and a substrate will come along that fits into it and bind to it. So once the enzyme binds to the substrate, a chemical reaction will occur between the molecules in the substrate and the enzyme, and then the enzyme will ultimately release the incoming substrate as a product. So usually two products will form. And in the bottom here, we can see the steps of an enzyme catalyzed reaction, and the specific example here we can see involves the enzyme hexokinase, which binds glucose and ATP. And the products are glucose phosphate and ADP, which are released from the enzyme. So as we go through the steps, we can see that the substrate in this case is ATP and glucose, and they bind to the active site. And once the enzyme undergoes the conformational changes stated by the induced fit theory that we learned before, and the enzyme will bind even closer to the substrate, and then the substrates are converted to their products, and then they are ultimately released from the enzyme. So the question here is, how can an enzyme speed up the rate of a chemical reaction? So let's take a look at activation energy. So when reactants come together to form a product, an initial input of energy, known as the activation energy, is needed to overcome their repulsion, which is formed by their electron clouds. So when reactants come together, an initial, which we can see in this diagram right here, an initial input of energy, which is the activation energy, is needed to overcome their repulsion. So the input of activation energy is necessary to reach the transition state so that chemical reactions can proceed to the formation of products. So in the diagram over here, we can see that the transition state is the peak, and the transition state is the state at which the original reactants bonds have been stretched to their limit. So once the reactants have reached the transition state, the chemical reaction can readily proceed to the formation of products. So ultimately, the activation energy needed to achieve the transition state is a barrier to the formation of products. So this is the reason why the rate of many chemical reactions are very slow on their own. Now, what enzymes do is they lower the activation energy needed, so transition state can be reached with only a small amount of heat. And enzymes do this by straining chemical bonds in the reactant molecules and also by bringing them together to facilitate the formation of new covalent bonds. Enzyme saturation. Enzyme saturation is when all of the enzymes within a solution are occupied with a substrate. Therefore, the addition of more substrates will not increase the rate of the reaction since all of the enzymes are already working. So let's take a look at this diagram right here. Let's see. Oh, sorry about that. Here we can see that the enzymes here are all have a substrate. So this would be representative of an enzyme saturated solution. But if an enzyme solution is unsaturated, meaning not all enzymes are occupied with a substrate, the addition of more substrates will increase the rate of the reaction because more enzymes can be working, of course, once they bind with another substrate. So in this diagram over here, we can see that the low substrate count means that this is a very unsaturated solution. And here it's more saturated. There are enough substrates to bind with each enzyme. And here, the high substrate count, which means there's a high substrate count, which means that there are way too many substrates and not enough enzymes. So this represents a very, very saturated enzyme solution, and the enzymes can't really deal with that. So those are the basics of enzyme saturation. Moving on to the role of inhibitors within enzymes. Competitive inhibitors are molecules that bind to the active site of an enzyme, inhibiting the ability of the substrate to bind with the enzyme. And their structures usually mimic that of the enzyme substrate. On the other hand, non-competitive inhibitors are molecules that bind to a location other than the active site of an enzyme, which is called the allosteric site, and they inhibit enzyme function. Allosteric activation of an enzyme can change the shape of an active site so that the enzyme can bind to the substrate and catalyze the reaction. So that is called allosteric activation, which is a positive effect, but allosteric inhibitors produce a negative effect so that they inhibit the binding of the substrate to the active site. So ultimately, allosteric regulation controls whether an enzyme is able to catalyze a substrate or not. So this diagram here shows an example of competitive inhibition. So the inhibitor would come along mimicking the active site of the enzyme and bind to it. Therefore, when the substrate comes along, it cannot bind with the enzyme, inhibiting that effect. Over here, let's look at non-competitive inhibition. So here we see the non-competitive inhibitor binds to the allosteric site, which is not the active site. Here's the active site, but it's binding to a site other than that on the enzyme. And therefore, it changes the shape of the active site so that when the substrate comes along, it's not allowed in. It can't fit. Now let's discuss the optimum conditions for enzyme activity. So enzymes are most active usually within a narrow range of temperatures. So many human enzymes are active at 37 degrees Celsius or 98.6 degrees Fahrenheit, which is body temperature. And optimum pH environments differ depending on the enzyme. So some enzymes prefer acidic environments, and others thrive in alkaline environments or basic environments. And ionic conditions also determine whether enzymes can function or not.